My name is Nurrahman Farurozi, but you can call me Ozzy. And what an amazing, precious change for me to be a master ceremony of this event guest lecture at Suraji Tirtonegoro Hospital in Clarence City on 21 November 2024. Perkenalkan, nama saya Nurrahman Farurozi atau biasa dipanggil Ozzy. Suatu kehormatan bagi saya pada kesempatan kali ini bisa menjadi MC pada acara kali ini, guest lecture di Suraji Tirtonegoro Klaten pada tanggal 21 November 2024. Once again, let's give a round applause for all of us. Before we begin, I'd like to take a few moments to explain the rules of today's event. The first one, sebelum kita mulai acara, izinkan saya membacakan sedikit peraturan pada kesempatan kali ini. The first one, please make sure your cell phones are on silent mode or switch off. So it don't disturb the speakers and other attendees. Mohon berkenan untuk dapat membuat mode handphone untuk silent mode atau dimatikan agar tidak mengganggu jalannya acara pada kesempatan kali ini. We encourage you all to participate in the discussion and ask questions related to the seminar topic during the Q&A session at the end of each session. Please make sure you keep your question brief and to the point to allow for a productive discussion. Kami menghimbau kepada Bapak Ibu sekalian untuk menyiapkan pertanyaan yang nanti akan disampaikan untuk sesi Q&A atau sesi tanya jawab untuk setiap sesinya. Dan kami harapkan pertanyaannya sesuai dengan tema agar diskusi kita menjadi lebih efektif. Third one, we request our attendees to refrain from any side conversation during the event as it can be distracting for the other participants. Kami harap kita di sini bisa mendengarkan dengan seksama, tidak dengan ngobrol satu sama lain agar tidak mengganggu partisipan yang lain. And the fourth one, please make sure you stay for the during of the event as it will provide a holistic understanding of the topic. Kami harapkan Bapak Ibu sekalian dapat mengikuti acara ini sampai dengan selesai agar dapat mengetahui dan menerima pengetahuan secara holistik untuk topik yang akan kita bahas kali ini. We hope that you will adhere to these rules and participate fully in the seminar. Thank you for your cooperation. Kami harap peraturan ini dapat ditaati dan terima kasih atas kerjasamanya. We would like to start this session by inviting everyone to stand up for the national anthem of Indonesia which will be played shortly. Sesaat lagi kita akan menyanyikan lagu kebangsaan kita, Indonesia Raya. Untuk itu mohon hadirin untuk dapat berdiri.
Di sana aku berdiri.
Thank you everyone, please take your seats. Hadirin kami silahkan untuk dapat duduk kembali. Now, let us hear welcoming speeches. First, please welcome Director Medical and Nursing, Mr. Widodo Wirawan. We would kindly invite it to the stage. Selanjutnya kita akan dengarkan sambutan. Sambutan yang pertama akan disampaikan oleh Direktur Medik dan Keperawatan, Dr. Widodo Wirawan MPH. Kami silahkan untuk memberikan sambutan. Please come forward.
Terima kasih, thank you Mas Ozi. Yang kami hormati Prof. Dr. Titi Nuryastuti, MSc, PhD, SPM, spesialis mikrobiologi klinik, konsultan dari Ketua Departemen Mikrobiologi FKKMK UGM. The Honorable Professor Christine Imber and Professor Estel from University de Poitiers. Yeah. I would like to say from to French, bonjour. Is right? Okay, thank you. Good morning and thank you for visiting to our hospital. Nice to meet you all. Dan segenap Bapak Ibu yang hadir pada hari ini dalam rangka guest lecture bersama dua profesor pakar dari mikrobiologi, parasitologi, dan tema kita nanti akan berkaitan dengan riset biofilm yang dihubungkan dengan jamur, kandida, dan infeksi penyakit kulit lainnya yang berhubungan dengan Malassezia. Saya kurang paham terus terang topik ini, but we are so much thanks to our expert from the France, both of you, the Professor Christine and Professor Estel, for as a speaker in our event here in Suraji Hospital. We are talk about the Java right now. Your first time visiting in Java, Professor Estel, yeah. You can take around to our hospital in Yogyakarta, especially in Klaten Regency, not Klaten City, but Klaten Regency, ya. Ya, jadi Bapak Ibu, dalam rangka peningkatan mutu di Rumah Sakit Suraji ini, kita memang terus-terusan untuk berkolaborasi dengan berbagai pihak. Dan yang paling lama sejak awal kita berkolaborasi dengan FKKMK UGM, khususnya dalam penelitian-penelitian terkait pengembangan layanan di Rumah Sakit Suraji. Nah, dalam hal ini biofilm ini juga termasuk kerja awal Bapak Direktur kita, our President Director, Dr. Solahuddin Ratomi, orthopedic surgeon, is one of the member of the biofilm research collaboration. Jadi saya sendiri bersama teman-teman direksi lainnya dan teman-teman yang berada di lembaga Diklit itu sangat mensupport adanya kegiatan ini dengan harapan kita semua bisa mewujudkan mimpi kita to be the leading hospital in Asia level. Yeah, this is the big dream, our big dream. Yeah, this is very challenge as a little hospital from the Klaten Regency. Ya, jadi Bapak Menteri kita itu mengamanahkan kepada kita untuk salah satunya menjadi research hospital. Ini tidak ringan karena kita saat ini bukanlah rumah sakit pendidikan utama. Yang pertama itu, yang kedua kita juga belum resmi menjadi penyelenggara hospital base for the resident education. So Prof. Christine and Prof. Estel, we don't have now to be the residential hospital for the specialists. We just only the satellite hospital for the specialist education. But in the future, our hospital will become the one of the hospital base for the conducting education for the specialist dokter. Jadi Bapak Ibu, untuk kuliah tamu pada hari ini, dari tamu expert kita dari Prancis itu akan berbicara tentang infeksi jamur, Candida dan Malassezia furfur yang nanti dihubungkan dengan sistem imun bagi pasien yang menjalani kemoterapi dan radioterapi. Ya. So tadi Prof. Titi bicara kepada kita tadi di ruangan merah putih, apakah di sini sudah mempunyai radioterapi? Ya, saya katakan kita belum mempunyai radioterapi, tapi kita sudah mempunyai onkologis radioterapi, Dokter siapa namanya? Dokter Luki. Dokter Luki baru saja WA-an dengan saya tadi malam. So Dokter Luki is only 1 hour oncologist radiation here. Temporary, he is existed at the Hussein Hospital at the Palembang City and Sumatera Selatan ya, betul ya, South Sumatra, in the Sumatra Island, not Java Island, ya. Yeah. So we don't have the radiotherapist module because we still wait for the groundbreaking for our cancer building in the past, the next month, I think the next month. We have the groundbreaking for the establish our cancer center. Kami mengucapkan sekali lagi kepada semua pihak yang sudah bersemangat, bekerja keras untuk mengusahakan kerjasama ini secara kontinu. Sekaligus saya mengaturkan permohonan maaf karena Bapak Direktur Utama kita, I apologize to you all because our President Director, the Suraji Hospital, cannot come here because he is, I think, he's on way home back to Yogyakarta. So I represent him to give the speech here. Akhir kata, last but not least, semoga guest lecture ini dalam rangkaian kegiatan visiting profesor mikrobiologi, farmasi dan parasitologi dapat menjadi sumber informasi yang bermanfaat dan inspiratif bagi kita, terutamanya kemajuan Rumah Sakit Suraji untuk mencapai rumah sakit unggulan di level Asia. Terima kasih sekali lagi Bapak Ibu semua, selamat pagi, Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Bonjour, Dr. Widodo, for the speech. Bonjour ya? Bonjour, okay. Correct me if I'm wrong ya. Okay, next we would like to hear another speech from head of the microbiology department of University of Gadjah Mada University. Kita akan mendengarkan sambutan yang selanjutnya dari Kepala Mikrobiologi Departemen Universitas Gadjah Mada, Profesor Dr. Titi Nuryastuti, MSI, PhD, Spesialis Mikrobiologi Klinik, Konsultan. Kepada Dr. Titi, you are welcome to the stage. Please give a round of applause.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. And yeah, good morning everybody. Thank you very much Mas Ozzy for this opportunity and distinguished guests, the director of Suraji Tirtonegoro Hospital and the staff, and also our esteemed speaker, Professor Christine Imbert, PhD, and Professor Estelle Perrault from University de Poitiers, France, honorable participant from today's guest lecture event. First and foremost, let us extend our gratitude to Allah Subhanahu wa Ta'ala for His blessing and grace that allowing us to gather here today for this important guest lecture event. So this program actually is a collaboration between the Suraji Tirtonegoro Hospital and also collaboration with the Indonesian Biofilm Research Collaboration Center with the Dr. Ratomi as well as the honorable member of the IPRCC. Actually, it is a remarkable initiative in advancing our understanding of microbiology, particularly in the field of biofilm and its clinical challenge. In this time, we are deeply honored and privileged to welcome to our standing speakers from University de Poitiers, Prof. Christine Imbert, that will discuss about the pathophysiology of biofilm associated infection, including the challenge of therapy. Yeah. And also Professor Estelle will present about the example of a real biofilm infection, focusedly on malassezia. So we hope that their presence will offer us a rare and invaluable opportunity to learn from internationally renowned experts in the field. Yeah, as we already know also that right now biofilm pose a significant challenge in medical practice. Yeah. Biofilm also give an impact in the antimicrobial resistance. Yeah. So the presence of biofilm will be challenged, and we should focus on diagnosis and also therapy. Yeah. As the head of microbiology department, I would like to express my highest appreciation to the director of Surajji Tirtonegoro Hospital for their hospitality, yeah, and also for all staff that initiating this event, also the member of the IPRCC organization. So we hope that this collaboration will support us and strengthen the synergy between academic, clinical, and also research, and we hope also that it will support this hospital to be a leading hospital in the field of research. Yeah. So finally, I hope this event will run smoothly, provide valuable insight, and also serves us as a foundation for closer collaboration in the future. Thank you very much, and Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you so much, Dr. Titi. And of course, ladies and gentlemen, before we go to the main event, there will be a photo session and handover of souvenir from Surajji Hospital to speakers and guests. So we encourage everybody participant to stand up during the photo session, please. Please stand up, please. Ya. Selanjutnya kita akan melakukan sesi foto bersama dan penyerahan souvenir dari Rumah Sakit Surajji Tirtonegoro kepada tamu undangan dan juga narasumber. Kepada seksi dokumentasi dan juga kamera, silahkan untuk menempatkan diri. We are going to take one video first. We can say hi to this young girl, young ladies. Okay, I'm counting one to three. So we are doing like this. Yeah. Okay. Okay. 1, 2, and 3. Okay. It's enough? Okay. Now, next, we are going to take a photo. Please, the photographer, come to the stage. Young ladies. Okay, please look to the camera. I'm counting 1, 2, 3. Give a very, very big smile. 1, 2, and 3. And now we are going to Surajji symbolic for photographic. We are going to
like this, Surajji symbolic.
Yeah, S for Surajji. So the right hand is up and in top. Yeah. Yeah, okay. Okay. Everyone's ready? Don't forget the smile. 1, 2, and 3. Please give us big smile. Okay, give a round of applause for all of you guys. And now we are going to hand over the souvenir from Suraji Tirtonegoro Hospital to guests and the speakers. So please come to the stage, the director of Suraji Tirtonegoro Hospital, Mrs. Dina, Mr. Widodo, and Mrs. Dewi. The first one, Mr. Widodo, please come to the stage and going to and to Professor Dr. Titi Nuryastuti, MSI PhD. Kami persilahkan Dr. Widodo dan juga Ibu Titi.
Maaf ini Prof Titi soalnya lagi direnov nih sempit.
Saya nggak usah dong, malu.
Enggak apa-apa. Ngadepnya gimana?
Hadapnya ke kamera. Nah, ya.
Maaf ya Bu, ini sempit-sempit.
We are so sorry for the space, but I hope we still can be enjoy the show. Thank you, Mr. Widodo and Mrs. Titi. And next, please come to the stage, Mrs. Dina and Professor Christine Imbert, PhD. Thank you. And we also invite the third one, Mrs. Dewi Anggraini, SAMM, and Professor Estella Braut Chatu. We have a journal of Suraji, so we hope that the journal is. Yeah, okay. The session photo is going to begin. We are counting to 3. 1, 2, and 3. Don't forget the smile. Thank you, Mrs. and Mr. Widodo and all of you guys. So we are so happy that we are going to the main event of this occasion. We've got an exciting lineup of speaker for today's event, of course, and will be guided by an amazing moderator. Pada kesempatan kali ini kita akan mendengarkan, kita memiliki line up atau memiliki narasumber yang keren yang tentunya juga dipandu oleh moderator yang luar biasa. We would like to invite him to the stage. Please welcome Mr. Emmanuel Billy Brilandi. This stage is yours. Please give a round of applause.
Okay, good morning everyone. My name is Billy and I will be your moderator for the session of guest lecture. So first of all, it's my honor to become a moderator today, to sit in front of you and to guide the guest lecture from morning to afternoon. Today we will have 2 lectures. The first one will be presented by Prof. Christine Imbert, PhD. The topic is about pathophysiology and therapeutic challenge in biofilm-associated infections. And then the second one will be presented by Prof. Estelle Perocato, PhD. The title will be Malassezia Associated of Biofilm Infection in Skin Diseases. So the rule of the lectures, it will be held around 30 minutes for each speakers. And then the discussion session will be at the end of both lectures. So we will begin from Prof. Christine and then Prof. Estelle, and then we will continue with the discussion session at the end of the lectures. Okay, without further ado, we will invite the first speaker, Prof. Christine Imbert, MSc PhD. Please come to the stage. You can go from here. Yeah. So before we go into the lecture, I will quickly read the curriculum vitae of Prof. Christine Imbert, MSc PhD. She is a full professor of parasitology and also medical mycology, School of Pharmaceutical and Medicine Science, University of Poitiers. And then she is also editor-in-chief of the Journal of Medical Mycology. So for everyone who submit to Journal of Medical Mycology, she is the editor-in-chief. And then she is also lead of the team microorganisms host environments of EPI lab. She did her PhD training in around 1992 until 1994 in the lab of parasitology and medical mycology, School of Pharmaceutical Science, Montpellier, France. And also she has a lot of publications and also research regarding the biofilm. I will not read all of them, and perhaps a few of them. The first one is screening antibiofilm activity of invasive plants growing at the slope Merapi Mountain, Central Java against Candida albicans, and also can leaves and stems of Rubus idaeus L handle Candida albicans biofilms. So mainly about biofilms and Candida albicans, right, Professor? So without further ado, now we will begin the lectures from Prof. Christine Imbert, PhD. Please give applause for her.
Can I have, please, the pointer? Hmm? Ah, yes. It doesn't work. Oh, yes, thank you. And can I keep the... To point? Ah, no, I don't understand how it works. Okay. How to point? Excuse me, Titik, do you know how it works? Oh, yes. Okay.
Yeah.
I have
And
to
then, up and then previous.
Oh, okay.
Yeah.
Okay. Thank you very much. So, good morning, everyone. It's my great honor to be there. Thank you very much for the opportunity to do this talk. First of all, I have to say that I am not a physician, I am not a medical doctor, so my point of view and my presentation is that of a researcher, so a senior researcher, but not medical point of view. So it's important. So my goal is to give an overview of the pathophysiology and the therapeutic challenges of biofilm-associated infections and especially of biofilm-associated candidiasis, because Candida is my main model. So I would like to start by introduce briefly Candida. Candida albicans, with its, it's not a car. Candida albicans is not a car. So Candida albicans is a member of the kingdom fungi. It can adopt different morphologies, different forms, depending on the status, depending on the environmental conditions it has, and especially you can have the yeast form and different types of hyphal forms, pseudo-hyphal and real hyphae, that can be short or longer. So it can adopt different morphologies, and some of them, especially, this one is very important in the infection process. So I will come back on this morphology later. So also to introduce Candida albicans. Candida albicans is a commensal. First of all, it is a commensal. So that means that in most part of human, it acts as a commensal of oral, digestive, and genital candida. So it's normal to find candida in human body. And unfortunately, in some cases, it can change its behavior and become an opportunistic pathogen. So it depends on the immune system, independent of the status of the patient. So, and in this case, when it is an opportunistic pathogen, it can be involved in infections that can be single species or polymicrobial. And there is a very wide range of severity of the candidiasis, as we can have mucocutaneous, superficial, or invasive levels. According to the CDC data, Candida species, not only Candida albicans, but Candida genus is the fourth most common cause of nosocomial sepsis. And finally, what is very interesting for me and for my team is that Candida albicans is a very good biofilm former. So that's why I speak of Candida today. So let's move a little bit to infection, and we have to understand that adhesion is a prerequisite for the infection. So that means that first the yeast have to adhere to something that can be a biotic or a abiotic substrate, and then we have the colonization step. And once the colonization step is succeed, we can have infection. And there are two different cases. The infection can be induced directly from colonization, or the colonization can promote the formation of a biofilm, and this biofilm can promote infection. So that means that the infection can be directly or indirectly induced after colonization. So, what is a biofilm? A biofilm, according to the definition of a biofilm consists in a community of microbes that are linked together and to a substrate. And this substrate can be inert or not inert substrate, can be solid or not, it can be a mucus, it can be different types of substrate. But what is important is that it involved receptor-ligand links, and that the attachment become irreversible. Also very important, there is a matrix. All these microbes in biofilms are embedded in a matrix, and this matrix is produced by the microbes themselves. So you can see here a beautiful picture that we took in the lab a few years ago, showing a Candida albicans biofilm. And so you can see here the yeast form, yeast morphology, and this is the matrix. This is the matrix, and in fact, at the beginning, it embedded really the cells, but because of experimental difficulties, when you take photo, it disappears. So it becomes like that. So microbes can adopt two different way of life. One is the planktonic way of life, sorry. They are free floating, and this is an artificial way of life, a lifestyle. It's artificial. It's the lifestyle we find in our labs, in our test tube, in Erlenmeyer, but not in the real life. In the real life, microbes mostly adopt the sessile mode of life, which is the attached lifestyle. So they form aggregates, and these aggregates are biofilms. So depending on the size and on the features of the aggregate, we can speak about aggregate or biofilm, but it is the same thing. So you all know a lot of biofilm in your environment. So of course you can see the boat surfaces. This is a beautiful biofilm, but you can see also the joints of a washing machine, siphon, shower heads, tubes and lines, and there are a lot of biofilms in our environment, and of course we have also a lot of biofilms possibly in our human body. So in the most well-known biofilm is the plaque, the dental plaque, and this is a very complicated, complex biofilm, as it can host about or more 500 species. So it's a huge and complex biofilm. So everywhere in the body, biofilm can form, and you have in blue all the places where a biofilm can form associated to a device, and you can see some catheters, and especially vascular catheters, peripheral vascular catheters, central vascular catheters, and you can see in red or pink the places where biofilm can form directly on the cellular surfaces. So, of course, you can find chronic wounds, endocarditis, kidney stones, and so on. So, what's wrong? What's the problem? What's the issue with biofilms? Actually, the microbes that adopt this the biofilm lifestyle become less susceptible to both antimicrobial agents and immune factors, immune defenses. So infections related to biofilms are very difficult to treat, or at least more difficult to treat than those that are not associated with a biofilm. So this is the first point. And the second point, and it is really important to take into account, and it will be one of my main message, is that there is an aggravating factor, a possible aggravating factor, which is that the biofilm can host one species alone, but sometimes they can also host other species. And we have polymicrobial B species or more polymicrobial biofilms. And you will see later at the end of my presentation that this is an aggravating factor, because they protect, each species can protect the other, and so biofilm become more difficult to treat. So let's move to the pathogenesis of candidiasis, and here of invasive candidiasis. So this invasive candidiasis can have different origins that are shown in this on this cartoon, and this comes from a manuscript from Pappas and collaborators. And first of all, keep in mind that in normal conditions, Candida albicans is a commensal, so it can be detected on the mucosal surfaces of about 50 to 70% of human as a commensal. But when it becomes pathogenic, we have two different cases, and the first one is on the left, is here, and actually, in some cases, after a especially after a gastrointestinal surgery, Candida can disseminate to the abdominal cavity directly into the bloodstream and induce a candidemia. And the second case, so this was, this was the case of colonization induced directly infection without the biofilm step. And the second case, the colonization can induce the formation of a biofilm, which can induce the infection, correspond to certain cases, especially where our immune response is decreased or because of other factors. And in that case, the Candida cell can produce hyphae. So hyphae, I said previously, are the invasive forms. And so the hyphae go to the bloodstream, and in the same time they form biofilms. And so in that case, we have the biofilm, which is an infectious reservoir, and some yeast can detach from this biofilm and go to the bloodstream, inducing the infection, the candidemia. So, as previously said, catheter, and especially vascular catheter, can be important substrates for the biofilm formation. So, CVC, central venous catheters, are very important device as they are at high risk of biofilm development. So I would like to move to this important point. And generally, in that case, the catheter-related bloodstream infection generally emanates from either the insertion site of the hub, or insertion site, sorry, or the hub, and involve the biofilm formation. The origin of, we are in that case different possible origin of the yeast, and this is what this slide tried to explain. So in case of intraluminal route, the yeast migrate along the inner surface from the hub, or sometimes, but it's really rare, they can come from a contaminated infusate, but it's not the frequent. It's not frequent, of course. In case of extraluminal route, the yeast migrate from the outer surface of the catheter, from the skin of the patient himself, or from the medical staff. And finally, as previously said, we can have also the hematogenous route in case of dissemination of the yeast after a gastrointestinal surgery. Oh, sorry. So a few words to show you the importance of this biofilm for the mortality, for example. So the biofilm-related candidemia are associated with an increased mortality. As you can see, based on the data of a meta-analysis of a Spanish team published in 2022, so quite recent, they observed that the mortality rate of candidemia would be about 40%. And if they consider only the fatal cases, they observed that 70% of the fatal cases involved a biofilm. So that shows the importance of biofilm and the consequences, the possible consequences of the presence of a biofilm in the life of the patient. So this confirms that it is a major issue in the pathophysiology of candidemia. So based on the knowledge of all these obstacles, a lot of groups have decided to publish guidelines. And this slide shows you the guidelines published by the HuskMID biofilm group, ESGB. So they are quite old because they are 10 years old. But actually, they are nothing new. They are still updated. There is nothing new to change. The people who do these guidelines thought 2 years ago it was needed or not to update them, and decided no, it's not necessary because there is nothing new. So we can still consider these guidelines are updated. So the main messages, the main points I would like to highlight from these guidelines is first that antibiotic and antifungal agents susceptibility testing of biofilm-living microbes is not predictive of therapeutic success. So that means that if we do not know if the microbes are in biofilm or not, the MIC meaning is questionable. You know, if it is not in biofilm, of course, it's good. But if it is in biofilm, in human, in our test tube, it will be not in biofilm, and the results will be different. The second point I would like to highlight is that the catheter removal and systemic antimicrobials are still the cornerstone of catheter-related bloodstream infection treatment. So this was a very important point of these guidelines. Finally, also in these guidelines, the important message was that the removal of the catheter is still recommended, is always recommended when feasible, of course, because all patients, it's not possible with all patients, but if it can be removed, it is better because the treatment of the biofilm on catheter is very difficult. And about and this is especially for candida infection, it's not for bacteria. And finally, about the treatment, the drugs for candida biofilms, what has been shown by numerous authors in vitro and in vivo research is that a few antifungal agents are active, or at least partially active, against biofilms of candida, which are lipid form of amphotericin B. Amphotericin B is not really active, but lipid forms of amphotericin B are. And echinocandins such as caspofungin and micafungin and a new one are active, have demonstrated antibiofilm activity, but none is completely active. So to end this presentation, I would like to come back to one of the first message as said, as the polymicrobial nature of a biofilm can be an aggravating factor in the disease. So what about the polymicrobial? You can see in this cartoon that we have different colors mimicking different species of microbes. And when they live together in a biofilm, I said previously that they interact thanks to direct interaction, physical interaction. So that means specific links, but they also interact indirectly thanks to molecules such as chrome-sensing molecules, such as metabolic product molecules, and so on. This is the indirect interaction. So we have both indirect and direct communication between the microbes. And this communication can be both between a single species biofilm and between different species of a polymicrobial biofilm, of course.
10 minutes left, Professor.
Sorry?
10 minutes left.
Yes, thank you. So, based on this communication, we can have different relationships that can be established, and sometimes we have antagonistic relationship. So in that case, I will not give details, but for example, when you have Candida albicans and Pseudomonas aeruginosa in the same biofilm, the relationship is antagonistic, and in fact, it promotes Pseudomonas aeruginosa, but not Candida albicans, and Candida albicans at the end is dead. It's used by Pseudomonas. Sometimes we have additive relationship, and sometimes we have synergistic relationship. That means that the fact there are more than one in the biofilm help each one to fight the antifungal drugs, the antibiotic drugs, the immune defenses. So this case is a problem for treatment. So in this case, in a case of a synergistic relationship, it is an aggravating factor. So I will give two examples. The first one is based on the PhD work of a student, Clément Bernard, who did his PhD in my team, and he studied the biofilm formed by Candida albicans and Cutibacterium acnes, the bacteria Cutibacterium acnes. And what you can see here, some pictures showing this B species biofilm. So you can see the biggest that are Candida blastospores, yeast form, here a hyphae, and the smallest are Cutibacterium bacteria, and here again. So what did he show? You can see here on these histograms that in gray, in two gray, so in deep gray we have the treatment, and here the control, on the left the control. And so what is, he compared the activity of micafungin, so micafungin very active against Candida albicans. He compared the activity of micafungin against Candida albicans when Candida albicans was alone, single species biofilm, and when Candida albicans was together with bacteria, B species biofilm. And what you can see is that micafungin is the activity of micafungin is significantly lower in case of a B species biofilm. So you see that the bacteria can protect the yeast inside biofilm and make the antifungal agent less active against Candida. So you can see here that it is an aggravating factor. And in fact, how do they manage? We have observed that in fact Cutibacterium acnes is an anaerobic microbe, and Candida albicans is a aerobic one. And so locally, the yeast consume oxygen and create some spaces without oxygen that are very good for the bacteria, so bacteria can grow, and it is promoting, the presence of Candida is promoting. Okay. And my second example is about the bee species between Staph aureus and the Candida albicans. So we observe the same. This is the work of another PhD student, Guillaume Amion, you can see here. And this PhD student studied the bee species biofilm formed by Candida albicans plus Staph aureus. And here again, he tested the activity of the echinocandin micafungin against single species Candida albicans biofilm. Here, control without treatment, here with micafungin, and compared this very good activity, you can see in single species condition 80% of activity, and compared this very good activity to the activity against Candida albicans again, but in presence of Staph aureus. And you can see that we never obtain a good inhibition in case of the presence of Staph aureus. So here again, we observe that the bacteria, even if they are not susceptible to micafungin, are able to protect Candida yeast from the activity of micafungin. So here you can see a biofilm of Candida albicans bigger with hyphae and the small bacteria of Staph aureus. So my take-home message. My take-home message is first that for diagnostic and treatment, I think I have enough repeated that it's essential to take into account of the possible presence of a biofilm that in case of a candidiasis, and especially in case of an invasive candidiasis, as the biofilm is really involved in the pathogenesis of candidiasis. And it is also essential to be aware that the biofilm can be polymicrobial, so the biofilm can host more than one species, because these different microbes can protect each other. The two current major barrier we have, is that we miss some routine tools for diagnosis biofilm-related infections. It's the observation, the evaluation of the biofilm is not adapted for routine diagnosis. It's a research. Aspect. So we would need some easy, rapid, fast, and not expensive tools for diagnosis the presence of a biofilm. And the second barrier is that the current treatment we have, the conventional antifungal agents, are still not completely active against biofilm form, against sessile microbes. So we need more active molecules. And this is part of the work we try to do in our research lab, to find new molecules able to fight this biofilm. So I would like to say terima kasih to all of you, and thank you for listening.
Thank you so much, Prof. Christine Imbert. Merci pour les cours. Okay, thank you. You have your seat again. And ladies and gentlemen, please be patient and keep your question, because we will move to the second lecture of today's guest lecture. It will be provided by Prof. Estelle Perocato. Please welcome Prof. Estelle. Give applause. Okay, are you ready, Prof. Estelle? The time is around 30 minutes. Okay, thank you.
Thank you. Good morning, everyone. Thank you for giving me the opportunity to present you one of my favorite topics. So now, after Candida, let's talk about this unknown yeast named Malassezia. I will present you different features between clinicals to diagnostic. And first of all, I would like to introduce me and tell you that I'm Estelle Perrault, and I'm a medical biologist in the hospital of Poitiers in France, and also a teacher of parasitology and mycology at the medical faculty in Poitiers. And of course, do my research in Christine Lab at EBI. So, first of all, for Malassezia, I would like to tell you that it was first described in 1846 by a man called Carl Ferdinand Ehstedt, and it was isolated from Pityriasis versicolor lesions. First, it was named as Microsporum, and it was then renamed as Malassezia in honor to Louis-Charles Malassez. Then Mr. Saboureau gave them the name of Pityrosporum in 1904, and it has to wait until 1984 for them to have again the name of the genus Malassezia. First of all, until 1990, only two species were known, Pfeffer and Pachydermatis, and the species description have been improved by molecular tools, and to date there's about 18 species that have been described, and they have been classed in 3 clades, as you will see on next slide. Some species have a large host spectrum, you will see on next slide. They can be hosted by human, of course, but also by animals and different types of animals, always hot-blooded animals. And there exist also some specific Malassezia to only one animal or only human. So here a representation of the different species described at now. At the top you've got human species that can also be recovered, as you see here, on different animals. And their first identification date here. And I've represented to the clade they belong to. And here, the twin that they can use, which you can see later, is a part of a key of identification, or was a key of identification. At the bottom, you've got the non-human species that can be rabbit or parrots or bats, and that have been more recently described, as you see here. And of course, the most known species as Pachydermatis, Sympodialis, Globosa, Optosa or Restrictor are human species. But veterinary are also really interested in this subject. That's why new species are often described and newly described. So let's go now on Malassezia features. They are small yeast, smaller than Candida as described by Christine just before, and they belong to Basidiomycota. They are a commensal of the human skin, of warm-blooded animals, and human, of course. And as you can see here, their shape are ovoid, globose, or cylindrical. The typicity of Malassezia is the fact that the budding is unipolar and has a broad base that is typical of this genus. Some species can also form hyphae. That's the case of furfur, that hyphae can also be seen on the skin, on the lesions, not only in culture. These yeasts are also lipid dependent. They don't have, they lack fatty acid synthase, and they can't synthesize de novo fatty acid, so they need a lipid source to grow and to develop, and that's why they prefer some localization in human that are rich in sebum or oily zone, and in culture, in vitro culture, of course, this will influence the medium that you may use, because conventional medium does not contain enough oily compounds, and you must use mediums such as Dixon medium that contain twin and olive oil and favor the development of Malassezia. It has long been thought that Malassezia pachydermatis was not lipodependent because it was able to grow on Saburo dextrose agar, but recently Wu and his team have sequenced its genome and proved that the fatty acid synthase was missing too in this species. So now we know that Malassezia pachydermatis is not lipid-independent species, but it has been explained by the fact that it is able to use peptone that is contained in Saburo dextrose agar. So it's another way to obtain fatty acid, and it's not a question of different genomic. Okay. Let's talk now about the presence of Malassezia on the skin, on the human skin. So as you know, skin microbiota is composed of bacteria, fungus, and viruses too, which are commensal of the skin. And a recent study performed by Findlay and collaborators has shown that, of course, bacteria are largely predominant, but Malassezia is the most prevalent eukaryote present on the skin, on the human skin, in fact. And here they are represented in purple, and as you see here, it's largely represented as a eukaryote on the skin. It has been recovered on all body sites except foot, because of lack of sebum on the foot, probably. And it has been shown too that colonization by Malassezia starts just after birth. Of course, it increases during puberty and when sebaceous glands are in action, and it can increase until adult age, but it depends on the presence of sebum at the age between two people, okay? Depending on the studies, you will see, if you're interested in this subject, that there is a big difference in species described, because some species are really difficult to grow. So if the study is performed by culturing, some species will be identified, whereas if the study is performed by molecular tools, other species will appear, if I can say that, and it's because this difficult to grow species can be evidenced by another way than molecular tools. And to date, Restrictor globosa seems to be, using molecular tools, the most predominant species, far from Sympodialis. And notice too that Pachydermatis is often recovered in human, even if it's largely zoophilic species. And this is explained by the fact that it's present on different animals, and mostly dog and cats, and it could colonize skin of pets' owner. So as you've seen, Malassezia is just a commensal, or can be only considered as a commensal, but because of host and environmental factors, it can cause also diseases and disorders. In these diseases, we can talk about pityriasis or seborrheic dermatitis and dandruff, Malassezia folliculitis, atopic dermatitis, in which Malassezia is more and more involved, or supposed to be involved now, psoriasis too, but also, and I will not speak about a lot of that, but also systemic infections. So if we focus first on pityriasis versicolor, named also Tinea versicolor, it is present worldwide. Of course, the prevalence is really different between the localization and the populations, and it can reach a half population in some tropical countries, whereas in some cold countries only a little part of the population is touched. It is, as I say, most common amongst adolescents and young adults because of the sebum production, and it is not transmitted by interhuman contamination. The most famous species are Furfur, Globosa, or Sympodialis in pityriasis versicolor, and what we know is recurrences are frequent. Here I represented the different factors that could influence the becoming from commensal to pathogen, and you can see that it depends on the environment, but also on host factors, oily and lotion or cream used on the skin, excessive sebaceous secretion or pregnancy, the use of different medicines, as oral contraceptives or corticosteroids, or different diseases, as you can see them here. And pathogenicity is induced by the fact that the saprophytic yeast, or the budding foam, is able to become pathogenic by hyphoform present in the stratum corneum. So, clinically, pityriasis versicolor is evidenced by erythematous demarcated round or oval macles with thin scales at the surface, which can coalesce, as you can see here or here, into patches. Hyperpigmentation give them the name of pityriasis versicolor, and the lesions are mostly located on areas which are rich in sebum, and this consists of the upper trunk, so the neck, the shoulders and upper arms, and when the face is involved, it usually touches the forehead. The use of wood lamp is also a way to have a diagnosis, and it shows gold yellow, yellowish green, or coppery orange fluorescence depending on the lesion and on the species on the skin. For the seborrheic and dermatitis and dandruff, of course, you might know it. It's a scaly and erythematous state of the scalp that can be recovered also on the face, on the upper trunk and flexure, and frequently recurrent here too. The prevalence in general population is between 2 and 11%, depending also on countries and on the climates. And it seems to be more common, three times more common in men than in women. It's also responsible for cradle cramp in infants, also named at that. The predisposing factors are cold or dry climates and hormone levels. Of course, puberty here has an influence too. But immunodeficiency have a role in this one, and this type of lesions are often recovered in HIV patients. When it touches the scalp, seborrheic dermatitis is called like that when you accumulate scaling and inflammation. When there is only scaling, that is called dandruff. Next lesions are folliculitis. It's a type of inflammation of the pillar sebaceous unit because of the presence of a big quantity of yeast in the follicle, and this provide dilatation of the follicles. This leads to lesions which are erythematous, itchy, and present mostly on the chest. And predisposing factors are also hot and tropical climates, as in pityriasis versicolor, and high temperature and immunodeficiency. The species usually involved are globosa, restrictor, and sympodialis. Now, just a little talk about systemic infections. They touch immunocompromised patients and neonates, often preterm infants, who receive lipid supplementation or parenteral nutrition via catheters. The catheter colonization leads to bloodstream infections that could lead to deep-seated infections, and that can be possibly fatal. The most frequent species are Pfeffer, Pachydermattis, and Sympodialis. Be careful, because if you look after yeast on catheter, you might find Malassezia, and the presence of Malassezia on catheter does not obviously mean bloodstream infection or deep-seated infections, so it could only be colonization and can be solved by catheter removal. So now for the diagnosis, as I said, typical round lesion and scaling macules can be evidenced by wood lamp, and you can see here an example of, it's not evident here, but yellow lesions. And when you use direct microscopy using a dermoscope, it is possible also to evidence the alteration in the pigmentation and also the contrast in halocene. And it is possible to see mycelium fragmented into short filaments and thick-walled yeast, but also oval yeast alone can be seen. The lesion can also be evidenced by tape stripping, scale scraping, or swabs. And of course, direct examination is only performed on type stripping and scales. For scales, it needs to be pre-treated by potassium hydroxide to see on the microscope, and it can be necessary to use a dye too, whereas lactol phenol blue or calcofluor white. And in pityriasis versicolor, you can see has here clusters of small rounds ovoid yeast with sometimes short hyphae. And in seborrheic dermatitis, often it's only unipolar budding yeast. In folliculitis, clusters of yeast totally fill the follicles. It is also possible to use contact plates that can be put directly on the skin and permit to perform the culture directly, but in this case, no microscopic evidence of the yeast on the skin can be done. Culture can be done on, as I said before, lipid-enriched medium, such as Dixon medium, or which can be on agar lens or plates, because, as I told you, Dixon medium contains oleic acid and twin. It can be joined to antibiotics to avoid the multiplication of bacteria present on skin and which are commensal too. Sometimes some of you use Saburo.
10 minutes left, Professor.
Thank you. Some of you use Saburo addition with olive oil to permit multiplication of Malassezia. It works also. Only Malassezia pachydermatis can grow on Saburo dextrose agar, so when you perform a microscopic examination of the colonies, when you see a shape that could remind you Malassezia, but it has grown on Saburo agar, perhaps you can think that it could be pachydermatis. Chromogenic medium are available in France, I don't know here. It's sold as a medium that can permit differentiation between Malassezia and Candida, and it contains antibiotics and inhibits bacteria growth too. Medium incubation needs 4-5 days at 27-30 degrees. In case of skin samples, when you perform blood culture, of course, they are done between 35 and 37 degrees. Creamy yellow to brown colonies are observed on standard medium, even if you use a chromogenic medium and they take a specific color. In the most famous medium used in France, they take a pink color, whereas Candida is blue. Colonies microscopic examination show the characteristic small ovoid yeast with broad-based unipolar budding, similar to a footprint. We call that similar to a footprint. As you see here, it's like a shoe with the top at the end. Here you can see a photo of microscopic electronic microscopy that we performed at the research lab, and you can also see the broad base budding which is typical of Malassezia here or here also. Be careful. Older species, as I said before, do not have the same requirements to grow in vitro. That's why Restricta and Globoso are quite fastidious to grow, even at 37 degrees, whereas Pachydermatis, Pfeffer, or Dermatis can grow easily up to 40 degrees. Histological examination can also inform us, and it can be possible to see a sort of, they call that spaghetti and meatballs, because you can see the budding yeast and the hyphae that are embedded in the stratum corneum. Okay? It took different coloration, and but mostly yeast-specific or fungus-specific coloration. for systemic infections, as I told you, blood culture are possible. It's not so easy because the medium does not contain oil, so a lot of Malassezia are inhibited in the vial, and it is supposed so that catheter culture would be better to guide to go to the road of a supposed invasive infection in Malassezia. It could be an information. Of course, you need to go further. Deep-seated infection can only be evidenced by biopsy culture. And to finish my presentation, the identification of this yeast is quite complicated, quite difficult. Of course, this identification has no impact on the management. It's only interesting for epidemiological data. Malassezia has typical characteristic of feature. Some of this feature has been used to identify them up to now. Malassezia has the other Basidiomycota urease activity, so using urea test, it's a way to think about Malassezia. Some of them, some of the specialists are able to distinct the species on morphology features. And in these features, different of them have been used to identify Malassezia before. Culture on Sabrodex agar permit to identify or to think into Pachydermatis. Catalase activity, they all have catalase positive except restrictor, but all the authors are not agree on this subject. Thermal tolerance can go up to 40 degrees for Pachydermatis, Dermatis, Furfur, Sympodiales of Slouflier. And as I have shown you on the first table, the use of the twin could be a good way to identify Malassezia, but it needs a good expertise, and interpretation discrepancies exist between authors. Chromogenic medium are validated for identification by mass spectrometry, Maldeturf. I don't know if you use that to identify yeast. Yes, and culture on Saboro plus olive oil also permits to identify them by this technology, but results mainly depends on the database that suppliers furnish you. And in France, in suppliers' database, there are only 2 or 3 species that can be identified using this method. So to date, molecular identification is the principal way to identify Malassezia species. It mostly can be, it is 1 or 2 EGH1, D1, D2 domains, or even demonstrated by some authors, beta-tubulin. So last slide to present you my take-home message. So Malassezia is a skin commensal, but also can be involved in diseases such as pityriasis versicolor, seborrheic dermatitis dandruff, and folliculitis, but also deep-seated infection and invasive infections. Biofilms can be possible on skin and materials in dandruff or in catheter. Of course, the infection deals with biofilms. 18 species have been identified up to date, but more or less cultivable between the species. The typicity of budding with a unipolar budding and a broad base is typical for Malassezia, and this yeast is lipodependent, so you need to use a specific culture medium and mostly Dixon medium. But remember that even in Dixon medium, the growth is fastidious and difficult to obtain. And identification is also difficult and mostly rely on molecular methods. Thank you for your attention.
Thank you very much for Prof. Estelle for the presentation. Now you can, and Prof. Christine can sit in the front because we will move to Q&A session.
Thank you.
So ladies and gentlemen, thank you for your attention during the lectures, and now we will begin the panel discussion. If there are any questions that you want to ask for both Prof. Christine and also Prof. Estelle, please raise your hand, and jika Bapak Ibu ingin bertanya dalam bahasa Indonesia, diperbolehkan atau dipersilakan, saya akan membantu menerjemahkan. So please, if anyone would like to ask something about the lectures today. Oh yeah, we have a lot of questions today. So firstly, maybe Prof. Titi. Oh, Dr. Fadila.
Mohon izin. Thank you for the opportunities. I'm Fadila. I'm from Clinical Microbiology Study Program in Gajah Mada University. I would like to ask to Prof. Christine about what the exactly mechanism from the amphotericin B and echinocandin as the antibiofilm in Candida infection. Thank you.
Thank you for the question. Actually, I will start with my response for echinocandins. So echinocandins, the mechanism of action of echinocandins, whatever the molecule, is that they act on a component of the cell wall of yeast. And so that's why it's quite normal they are active against biofilm, because the cell wall contains all the molecules, adenosines, and all the molecules involved in the direct link in the direct interaction between cells and between cell and substrate. So echinocandins target the cell wall of yeast. Okay, for amphotericin B, it's a very good question, because amphotericin B, it is known that it acts on ergosterol, but what it is not clear is that lipid form of amphotericin B are more active than conventional amphotericin B, deoxycholate amphotericin B. And this is certainly due to the ability of a lipid form of amphotericin B to go deep in the biofilm, so to go inside the biofilm. And I think that amphotericin B is not able to enter, to cross the matrix, to enter inside the matrix. In fact, one of the most important barriers for antifungal agents is the matrix, because the matrix is has many holes. One of them is that some carbohydrates of the matrix are like sponge, and they keep the antifungal drugs and prevent its action. I don't know if I answer to your question. It's okay.
Any more questions from Dr. Fadila, or is it enough? Okay, thank you, Dr. Fadila, for the question. And then for the next question from Dr. Konita. Oh yeah.
Hello, my name is Konita. Thank you very much for your presentation. It giving me much insight about biofilm formation, especially from fungal. And in here, I want to ask you, actually we know that Candida and Malassezia is our normal flora. and but in another hand, it can be pathogen, especially when the Candida and Malassezia making biofilm formation. In here, in that case, I want to know what is the strategy that we can use for prevent our normal flora like Candida or Malassezia to make biofilm formation, because we know that prevention is better than curation, and when biofilm is forming, it will give us poor prognosis for the treatment. Thank you very much.
I start and Estelle will continue. There are, yes, it's really also a good question, because they are commensal, and that's why we both start by saying they are commensal, and this is very important because that means that the status of the patient is the key point for me, and so there is no problem as soon as the status of the patient is good. So it's very difficult to do prevention. Prevention is to keep a good immune status for me. It's my general answer.
And to go further, we can also say that hygiene and in the use of catheter is mostly important for the invasive infections, and perhaps also that information is a way to act early in the infection. So cartography and knowing of the colonization is a way to act early in the infection and to avoid disseminated infections.
Yes, I can add that a quite recent study has shown that vascular catheters, in some cases, and quite often, were maintained in patients even if they were not needed anymore. And we know that these vascular catheters are very strong promoting factor for biofilm. And sometimes the physicians are not aware that there is vascular catheters in patients. This is the results of the study. So one key point of the prevention is to make the physician aware of the absolute need to avoid vascular catheters if they are not needed anymore.
Thank you very much.
Thank you. My name is Riana. I'm a dermatologist in here. So I would ask to Professor Christine. I saw in your slide that Corynebacterium acnes sometimes make a biofilm for protect the candida from treatment. So as clinician, should we treat the cutaneous candidiasis with antifungal combined with antibacteria such as clindamycin or benzoyl peroxide, because the Corynebacterium is an anaerobic bacteria? Thank you.
Yes. Again, a very good question, very complicated answer. And again, I am not a clinician, physician. So it's the point of view of the researcher that I will give you. The yeast and bacteria can interact, but what is the issue is that the diagnostic tools do not allow, in some cases, to highlight the presence of all the microbes that are really present in the biofilm. So, in fact, 2 points. It's not always possible to highlight the presence of a biofilm, so first point. So the routine tools highlight the infection, but not, we do not know if it is related or not to a biofilm. We have some idea, but it's not always obvious. And if the presence of a biofilm is suspected, if the presence of a biofilm is suspected, it's difficult to know if there is only one or more species inside the biofilm. So 2 points. Yes, of course, giving all the drugs together would be can be an option, but I think it's not the good one, because you will promote resistance, and you have to avoid absolutely resistance, especially as we know that resistance is a growing problem currently. So unfortunately, my answer is comfortable for the researcher, not for the doctor, but I would say we have to go as deeper as we can in the diagnostic, but not combine too much drugs, because even if it works on 1 or 2 patients, at the end it will not work at all for all patients. I don't know if Estelle, you share my opinion? Yes.
Thank you so much for the question from the participant. Are there any more questions? Prof. Titi.
Yeah, thank you very much, Prof. Christine and Prof. Estelle, for very inspiring lecture. For Prof. Estelle, I would like to confirm, as you said that malassezia can form biofilm on the skin, and is there do they consider as a good biofilm together with Staphylococcus epidermidis also on the skin, or yeah?
I suppose so, yes. In dandruff or in other types of lesions, we know that malassezia can form biofilms, and probably not alone.
Okay. Yeah, and then another question. So we could find malassezia also from blood specimen, yeah, because yeah, when we take the blood specimen, we do the invasive method, taking the blood, and so is there any consideration how to determine whether it is malassezia as a pathogen or just contaminant from the skin?
It's always difficult, but of course you have to combine clinic observation and blood culture results. And when the patient is suspected of fungemia or a sign of septicemia, then sepsis, then you must consider malassezia in this case, I think.
Okay.
If you have no other pathogen, it's mandatory to think of it.
Oh, okay. Yeah, thank you. Yeah, and the last question. You said that malassezia is considered as the fastidious fungi. Yeah. So can we grow this malassezia in a bactec or another plants?
We have just finished a little study in which we have inoculated vials with malassezia and fresh blood.
Yeah.
And for some species, there's no problem. It is detected until little concentration, small concentrations, it's even detected. Whereas for some other species which are fastidious to grow, even at upper concentration, it does not work.
So it depends.
It depends on the species.
Okay, thank you very much. Yes.
Thank you, Prof. Titi, for the question, and also Prof. Estelle and Prof. Christine for the answer. Are there any more questions from the participants that would like to ask to our beloved professor in the front? If you want to ask in Bahasa Indonesia, we will gladly translate it to English. Yes? Oh.
Thank for my opinions. I would like to ask, I am pulmonologist, and we often find that in sputum, many candida in sputum. And my ask, can microbiologic tests to differ about commensal candida and pathological candida? Because it is for me to treat the disease. Thank you.
Thank you for this interesting question. I used to say in France that really little number of cases exist of Candida pneumopathy, pneumonia. So we consider that when Candida is recovered in sputum, it's always a colonization of the oral tract, and that you can or may only treat when patient presents some clinical sign of neural presence, but not to treat pneumonia, Candida pneumonia. There's no test to distinguish these two forms, and really when you search on publication, there's a little case of Candida pneumonia, so I don't think so.
Thanks.
You're welcome.
So we have an online participant who asked for a question, Prof. Estelle and Prof. Christine. From Novi Perdana from pharmacy, she asked about tools to diagnose patient if to find infection-related biofilm. So, is there any tools? Are there any tools to detect the biofilm-associated infection? It's a very good question, but very hard to answer.
Here again, if there were a lot of tools, it would be easier to treat. Yes, in fact, there are some, depending on where the sample come from, so the distance, if we took different samples, from blood and from catheters, and if they are both positive. So if there are more than one site with positive results, this is a factor that are in favor of the presence of a biofilm-related infection. But there are some kinds of parameters like this one, but there is nothing concrete, more concrete. It exists some machine that try to evaluate the ability of the isolate to produce biofilm, for example, but it's never the biofilm directly sampled from the patient. So saying that, confirming that isolate is able to form a biofilm doesn't mean that there was a biofilm in the patient, is different. So if at my knowledge there is still no tool to really directly for diagnosis, for routine diagnosis, to confirm the presence of the biofilm. It's done in certain lab, such as Danish, some in Copenhagen. They do some, because they are expert in biofilm, clinical biofilm, and so they do research, they combine research and diagnosis. But it's not possible to do that at the level of every hospital. Estelle, you want to add something?
What is important? Is it really important to know if it's biofilm or not? In fact, what is important to know is it an infection, and if I want to treat it, what kind of molecule which I use? And I think that when you know that there is catheter or other materials that could be recovered by molecules, by yeast that are in biofilms, and that could probably be less sensitive to antifungals, then you should think, oh, I have materials, then I must replace it before treating, or I must put a treatment, replace it, and continue to treat. But the presence of the biofilm is not a key point, I think, on the treatment. It's important to know what you want to treat.
Yes, it's not important because we cannot see it. It's important, but it's not important as soon as we cannot see it. But all the recommendations, all the guidelines give you answer to this question. Because if there is a catheter, azole is not recommended at all. So if you follow the guidelines, whatever the guidelines, American guidelines, European guidelines, you will see that it is well mentioned that depending on the risk factors that are present in this patient, for example, the presence of a CVC, you have to avoid some antifungal agents and prefer others. So guidelines are very important to follow.
Thank you very much for the answer from Prof. Christine and Prof. Estelle. So I'd like to translate the answer. Jadi intinya untuk tools atau alat untuk mendiagnosis biofilm infeksi terkait biofilm secara direct atau secara langsung itu belum ada. Karena memang pada kenyataannya untuk mengambil beberapa, untuk melihat apakah isolat yang kita dapatkan itu memang memiliki biofilm atau tidak, itu cukup sulit karena tidak dilakukan secara rutin di praktek klinis kita sehari-hari. Dan isolat yang kita dapat, walaupun dia membuat biofilm, belum tentu dia memproduksi biofilm pada lokasi infeksi. Jadi yang terpenting adalah bagaimana kita kembali ke pertimbangan ke klinis saat mentreat pasien secara klinis. Kita lihat faktor risiko yang ada, serta jika memang ada device atau tools yang menempel atau ada di dalam pasien, kita ikuti guideline yang ada. Jika guideline-nya adalah meremove atau melepas kateter, maka mau tidak mau, sulit maupun mudah, itu mau tidak mau harus dilakukan. Karena kemungkinan besar pasien-pasien seperti itu memang terinfeksi biofilm. Dan sebetulnya jikalau ada infeksi biofilm maupun tidak, itu bukan poin utamanya. Poin utamanya adalah apa yang akan kita berikan kepada pasien. Apakah jika itu memang infeksi biofilm, obat yang kita berikan akan masuk ke dalam biofilmnya atau tidak, itu menjadi pertanyaan yang sulit dijawab. Sehingga poin pertama adalah kita mau tidak mau harus melepas device yang ada. Tapi nanti jika ke depan penelitian biofilm in research develop in the future, and we can directly see in between the biofilm and non-biofilm, and then we can treat directly, it will be very useful. Thank you very much. We have 10. Sorry, I would like to confirm if we use, if there is a CVC in the patient, we cannot use antifungal therapy. Is that what you mean? Was that what you mean, or?
You mean we cannot do antifungal therapy if there is a CVC in the patient? Is this? No. No. If there is a CVC in a patient, you have to shoot a specific antifungal agents. I mean, azole is not recommended at all because we know that it is not active at all on biofilm microbes. So if there is a CVC, you have to choose lipid formulation of amphotericin B, if you have, or echinocandins.
Ya, oke. So, jika ada infeksi fungal yang terkait dengan CVC, maka tidak semua golongan dari antifungal bisa diberikan, khususnya hanya golongan echinocandin atau amphotericin B. Ya. Azole tidak dapat diberikan karena kita perlu antifungal yang dia mengandung lipid. Okay, and we still have 5 minutes to discuss about all these fungal things. So if there are more questions to come, please ask, and Professor will gladly answer your questions. From Zoom, apakah ada lagi, Pak? Okay, we have one question more from Zoom. The question is, biofilms in chronic and acute wounds, is the approach still the same? If different, what points should be emphasized in treating the biofilms in acute and also chronic wounds?
I am less expert in chronic wounds since, in fact, in the case of biofilm that grows attached to a CVC or to a catheter, it is an acute infection. The biofilm is the reservoir, and from this reservoir, some microbes can detach and induce infection. In the case of chronic wounds, it's quite different because the biofilm is both reservoir and infection. It's not only the detachment of microbes that is responsible for infection. The chronic wounds itself is infection. So the mechanism of the pathogenesis of chronic wounds infection is quite different, and I am not expert. But yes, of course, the approach is the same as diagnosis is needed, and then treatment according to the species that have been diagnosed. Candida is not fine. It can be found in chronic wounds, but it's not a main species found in chronic wounds. Pseudomonas aeruginosa and are mostly involved together with other species, but yeast are not so much involved, so it's difficult for me to answer. Yes.
Okay, thank you, Prof. Christine, for the answer. I think the time is now up. Thank you so much for our professor today. Please give applause for Prof. Christine and also Prof. Estelle. And now I will give back the time to our Master of Ceremony. Thank you.
Once again, please give a round of applause for our moderator and our speakers today. And now, we would like to say thank you for all of you that participate this event until this time. And that's all. Sorry for anything that maybe not fit in you, or something that really not good in you. And That's all for our meeting today. After this, we are going to a hospital tour to Suraji Tirtonegoro Hospital with 2 cars over there. But before we going to hospital tour, we are going to take a photo first at the front of the office. So that's all for me. Have a great day and wassalamualaikum warahmatullahi wabarakatuh.